Hi everyone, let's talk about how to do this worksheet where we're going to learn to decode the mRNA code. So as a reminder, mRNA is the messenger that gets DNA's message out of the nucleus. That message can be decoded using this because DNA contains instructions to make the body. The body is made out of protein. Protein is made out of amino acids. These amino acids are what build the proteins the body is made out of, and different orders of amino acids make different proteins. So it is important to understand, though, if you're going to use this chart to figure out what kind of amino acids are going to the proteins, it's important to know this chart is only valid for a code in mRNA. As a reminder, RNA letters are read in codons of 3, and every 3 RNA letters is code for 1 amino acid. If you have many amino acids, they come together to form a protein, and protein is what most of the body is made out of. So, if, for example, you have AAA, the way you read this chart is you take the first letter of the code, find it here. The second letter is also an A, and they come together in this box right here. The third letter, A, tells you which of these 4 to use. See how A is the top right here? Follow it over. It's lysine. That's why I wrote lysine here. You can simply use the 3-letter abbreviation, but LYS is the abbreviation for the amino acid that this RNA letters codes for. These 3 RNA letters UUU would code for, see, U and U come together in this box, and then U is here, second from the bottom, so it's specifically PHE, which happens to be the abbreviation for phenylalanine. And then GUC, G and U come together here, and C is this one, is valine, VAL. CAG, C and A come together here, and then G is second from the top, so it's glutamine, GLN. AUA, A and U come together in this box. A is isoleucine, that's ILE. CUC is the next one. C and U come together here. C and U, and then the third letter is C, so CUC is this last one here. Leucine, LEU. UGC. U and G are the first 2 letters, so they come together here. And then C is this last one, so it's this particular one, cysteine. ACU, so A and C come together here, and then U is this one, that's threonine, THR. So once that's done, that's individual codons coding for 1 amino acid at a time. If you have more than 1 codon, here that's 3, 6, 9 letters, that's 1, 2, 3 amino acids. AUG is methionine. CGA, you look it up here, and that's this arginine. And GGC, if you look it up here, that's this one, the glycine, I believe. The next thing to mention is less RNA letters means less amino acids. More RNA letters is going to be more amino acids. So because this makes 3 amino acids, you're going to have 3 here, 2 amino acids. That's why there's 2 right here. AUG is methionine. A and U is here, and G is this one, methionine. And then I'll put a little dash connected to the next one. UCA is the next one, so I find U and C. And then A is this one, serine. CAG, UCA, and then CUU. CUU, okay, C and U comes together here, and then U is the last one, so it's this one, leucine. LEU. Okay, so notice, by the way, AGC and UCA both code for serine. That can happen. That's why you see amino acids come more than once on this chart. But that's 1, 2, 3 amino acids. So oops, put that in the wrong place. This one's only going to be 2 amino acids. There we go, that's better. And this one, if you do it right, should give you 4 amino acids. So I'll leave it to you, the viewer, to actually work these out, but they should follow the same format as this. All right, onward. The start codon. It says always begin reading mRNA sequences at the start codon. Look at all the examples in section 2 because they all begin with the start codon. AUG, 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 AUG. They all contain AUG. That's the start codon. Ignore all mRNA prior to the start codon. In the mRNA sequence below, circle the start codon. So if you go through and look, AUG, 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 you got to look for it and find right there. There may be subsequent ones, but only the first one is the start codon. The stop codons are these. Stop is not an amino acid, so there's no amino acid stop. It just means stop reading at that location. So that means when we give you something like this, you don't have to translate all of them. You only translate the ones starting with the start codon, and you stop as soon as you reach a stop codon. So you don't have to do the whole thing. I'll show you an example. But to recognize these, it helps to memorize them because then you spot them faster. So if you can remember you are great, you are awesome, you go away, then you know the stop codon. So it's literally just remember you are great, you are awesome, you go away. Helps you remember it. So write the start codon for practice, the AUG, because if you look at the start codon, it codes for methionine, MET. That means every amino acid starts the same way. They all start with methionine, no exceptions. All the proteins start with methionine as the first amino acid. Okay, so when you do an actual sequence, here's an example. This is how you're going to handle your homework. Look for the AUG and you circle it. That's where you start. And then you can start looking up on the chart. AUG comes for this. UUC gives this on the chart. CAC gives this on the chart. That's histidine, phenylalanine, glycine, whatever. So you go through, and when you find this one on the chart, UAA, UA, and then A is the top one, so stop. Stop is not an amino acid. Stop just means stop reading here, which means you can ignore all the stuff after the stop codon. So you do that, and then as you're doing that, you put these slashes in between every 3 to help keep it straight which one's for which. Notice that the amino acid is written directly below the codon. So I'm going to show you now how to do it for this one. And the first thing I do is I find the first AUG. So I'm just AUG. Oh, there's an AUG. So I ignore all things before, and this is where I start reading it off the chart. So AUG is methionine. I write methionine directly below, and then I'm going to start putting slashes every 3. See that other AUG? That doesn't mean start again. It just means it's another methionine. And so I'm looking how far I'm going to have to go. UCA, CUG, AGA. Oh, that's the you are great. That's the stop codon. So I know I don't have to do this for any of the others. I'm going to stop here because that's the stop codon. So now I look these up. CUA is, CU comes to this box, A is leucine. So LEU. MET, LEU. Putting the dashes between is helpful, not super required, but it is helpful. And then AUG is another methionine. GCA. G and C comes together here, and then A is alanine, so ALA. Notice how I'm lining it up. You need to do the same. And then UCA, so UC, and then A is serine. Alanine, serine, CUG, CU, C and U is here, and then G is leucine, LEU. Another dash, AGA, AG comes together here, and then A is arginine. So arginine, and then UAG is the stop codon, and I put nothing. Leave that blank actually, so I didn't even need to put that. So that's how you handle the amino acid sequence. So in other words, this RNA prompts the formation of this series of amino acids. This is the protein made by these RNA instructions. And because these are after the stop codon, you simply ignore them. Yes, there's another stop codon right here. Who cares? Ignore it. And down here, the process you follow is the same as up here. Now, something to consider. DNA is the master start sequence. If we give you DNA, you cannot read DNA off this chart. There's no T's. DNA has T's in it for thymine. So if we give you DNA, you're going to have to turn it into RNA first, then circle and put the slashes, and then look it up on the chart, just like here. Let me give you an example. We got to look for where it starts, and how do you know where to start? Well, you look for the tac. Why do I say that? Because T pairs with A pairs with U, and C pairs with G. So you put TAC in here, because DNA TAC is going to have RNA letters AUG pairing with it, and that's where you begin reading the DNA sequence. So here's what you can do. You can actually make it easier on yourself even than this. I'm going to go through here, and I'm going to find the tac. TAC. Oh, there it is. There's TAC. So this is the tac. I'm going to start translating. I'm sorry. I'm going to start transcribing here. Makes my life easier. AUG. U. CG. A. CG. U. AU. G. C. A. UAA, U, C, T, A, G, U, C, A. Now, as I look at these, I'm going to start now checking 3 at a time. 1, 2, 3, 4, and 3. All right, well, I don't know why I counted 1, 2, 3. So 3, 3, 3, 3, 3, 3, 3. Oh, this is the You Are Awesome. That's a stop codon. I don't have to do anything after that. So now, technically I didn't even have to write these letters, but whatever. It doesn't hurt. I don't need to put any more slashes because we're ignoring everything after the stop codon. Now I go look these up on the chart. We should know at this point, methionine is MET, it's AUG. They all start with methionine. U. U and U come together here, and then U is phenylalanine, PHE. So I put PHE, CGA, CG, and then A, arginine. And then CGU, so I'll look that up. C and G is come together here, and U is arginine. See, sometimes different codons can code for the same amino acids. That's fine. It can be 2 in a row. AUG is another methionine. Seen it so many times, can't help but remember. CAA is C and A come together here. CAA is glutamine, GLN. GLN. And then UAA is stop. So I just stop. And that's it. That's how you handle it. Make sure you have a slash between every 3 to show where your codons is. And make sure that you've written the amino acid directly below the RNA letters that code for it, the RNA codon that codes for it. And then that'll be that. That's what we need to see. So you follow the same steps here to do this particular one. All right, that's it. Have at it.